speaker, and I am honored to introduce the final speaker of this session, Dr. Pushp Sheel Shukla, a distinguished researcher and a leading expert in the field of biostimulants. With a PhD in plant molecular biology, Dr. Shukla brings a wealth of experience spanning over a decade. His groundbreaking work has been published in top-tier journals and presented at major international conferences, underscoring his significant contributions to plant science. As a senior research scientist at C6 Energy Private Limited, Dr. Shukla spearheads the development of innovative seaweed-based biostimulants designed to enhance crop growth, stress resilience, and nutrient efficiency. His work not only advances agricultural practices, but also contributes to sustainable farming solutions. Dr. Shukla's role involves deep research into the mechanisms of these biostimulants and their practical applications, aiming to elevate agricultural productivity and environmental sustainability. Previously, Dr. Shukla was a postdoctoral researcher at the Dalhousie University, where he focused on abiotic stress responses in plants, supported by prominent organizations such as the Natural Sciences and Engineering Research Council of Canada and MITAX Canada. His dedication extends beyond research. He's committed to nurturing future scientists and actively engages with both local and global scientific communities to drive forward the frontiers of biostimulant technology. Dr. Shukla's expertise in this field is truly transformative, and we are privileged to have him share his insights with us today. Dr. Shukla, may I now request you to share your screen and start your presentation, please? Thank you.
Thank you, Amalia. I think I'm visible. Yeah.
Yes, we can see your screen and hear you well. Yeah.
Okay. Thanks, Amalia, for such a nice introduction. I did not know that I had done so much of work. So, first of all, good morning, everyone from the easternmost part of the Canada. I live in Halifax, and I work for a C6 station in Halifax and supervise a team. And as my title mentioned, I basically worked in the area of seaweed biostimulants. And I assume this is a next generation input that helps sustainable agriculture. Yes. So most of the people think that when we are growing the plant is throughout his life cycle, they are confronted with the different abiotic stresses. Previously, people try to breed new varieties. They do genetic engineering and they try to confer stress tolerance. But with the new class of agriculture input, what we call a seaweed biostimulant, they try to improve plant natural processes and induce systemic tolerance and resistance in the plant. If I say a biostimulant, the literally meaning of the biostimulant, the compound, those biostimulant doesn't have any nutritional value of their own, despite they are derived from the natural, either it's a microorganism or the seaweeds, and they improve plant natural processes and help the plants to grow in the adverse conditions. My peers and my colleagues, Dr. Arup, they all mentioned a lot about the seaweed biostimulants. I will just try to put off more scientific on the plant science basis. So as we know, there's lots of discussion on this workshop. What are the seaweeds? Basically, they are the multicellular marine organisms. And they are the very good source of bioactive compounds because they live in the environment that is continuously changing. So they have to fight in that environment. They produces a very unique molecules that is helpful for their survival. And those seaweeds are either brown, red, or the green, depending upon the pigment they have. And if I say on the seaweed biostimulant, there are around 47 companies around the world that is producing those seaweed extract and commercializing those for the agriculture products. Their number might be varied, but I am okay. It's there. So different seaweeds have a different bioactive compounds, and as we know, the reason behind that because they grow in different environment, whether it is a green seaweed, whether it is a brown seaweed or the red seaweed. And those, if I give an example, alva has a different set of a carbohydrate called alvans. Carrageenan has a carrageenan, and asco has fullertannins and sterols. And these bioactive compounds plays a very specific role in inducing plant growth, and thus they improve the natural immunity. So um, I have worked in the last 10 years on the both the brown seaweed and the red seaweed extract. So I will just try to focus on certain snippets that come across that how about my R&D. So if I say brown seaweed based extract, mostly if I say 90% of the companies, they are using ascophyllum as a um, rhodosium as a raw material, and it is one of the most researched seaweed for the biostimulant production because it is a very rich source of phenolic compound called fluorotannins and has some unique polysaccharides like alginic acid, fucoids, and mannitol. And those initial 5 years of my career, I just worked on to understand how this brown seaweed helps the plant to improve their growth. And there are the other seaweeds also like Eclonia, Laminaria, and Sargassum. People are using them as also source of the raw material for the biostimulants. But uh, I have understand like how these seaweed extracts mitigate salinity stress in the plant. So in our lab, in the Prithvidas lab in Dalhousie University, Canada, we have shown that when we are applying these extract, the plants are surviving better in the salt stress. And there are various molecular and physiological mechanisms involved behind that. So if we see this heat maps represents the microRNA expression profile. So we can see there is a very distinct microRNA expression if we are seeing when we are adding ascophyllum extract as well in the presence of the salt. So I just summarize everything not to make it a complex. How does it happens? When a leaf surface has those biostimulant, it triggered a series of response that elicits both post-transcriptional as well as post-translational mechanism in the plant. As a result of that, there are certain genes which are called as an stress responsive genes are induced more, and it helps the plant to use the carbohydrate metabolism, cell wall biosynthesis in a more appropriate way in those stress as compared to the untreated plant. Similarly, we have shown the same kind of effect in presence of a drought stress also. Like when we are putting the brown seaweed extract in the plant, it changes certain physiological adaptations in the plant by regulating certain drought specific gene expressions and helps the plants to grow. So these are the examples that I just want to show that brown seaweed does in terms of stress tolerance. And but there is a limitations with the brown seaweed because most of the brown seaweeds are naturally harvested. They are not a sustainable source of a raw material, and too much of a harvesting of those seaweeds may have an ecological impacts too. And the other main limitation is that they are grown in the different geographies in the temperate climate, and the geography and the extraction procedures has an impact on their bioactivity compound. And the season in which you are harvesting those biostimulant also impacts the amount of the bioactive compound present in those seaweeds. So at the C6, that's why we focus on most sustainable source of raw material as a red seaweed. Right now we are focusing more on a Cupa alvarezii, and that is a very industrially important seaweed, as Dr. Arup Ghosh mentioned, and that is the main source of phycocolloid, carrageenan, and some quaternary compounds. So at C6, we have a subsidiary in the Indonesia and the Netherlands, and what we are doing, we are basically modernizing the cultivation of the Cupa on the ocean shore in Indonesia, in Lombok. We have developed 1 square kilometer of a farm using modern technologies, modern sensor-based technologies that provide us a sustainable raw material. And this raw material is grown in a very tropical zone with the almost uniform climatic conditions. So the raw material is almost uniform, and we get a very um, scalable raw material for the precision ocean agriculture. So I am not an expert in ocean agriculture technique. There is a very specialized team in the C6 that focus at that part of the cultivation, and we process that raw material. They give that raw material to me to make agrogen, AG4 and AG boost. These are some commercial biostimulant that we make from copper ficus at C6. And the main active compound of the copper ficus is carrageenan because it contains a D-galactose sugar. So there is a literature which shows that carrageenan itself has a plant growth promoting properties. So they are different from the form of the carrageenan, and because of their sulfation content, they induce different physiological and morphological responses in the plant. And this is reviewed by me in 2016 in Frontier Marine Sciences. So coming to my main topic of a talk, basically an agrogen. So agrogen is a biostimulant that is developed by the C6, and it has a patented active ingredient and a defined mode of action through the scientific literature. So I worked at the C6 as a senior research scientist to understand how the agrogen works in improving the plant growth. So what makes an agrogen unique? Because it has a patented active ingredient, and we have a differentiated products basically. It works at a very low dose, 100% natural and organic, and we have lots of data to support the scientific backing, how it improves the growth. Few of them I will discuss in coming slides. And it passes through a very strict quality control, both by using the chemical signature as well as bioassay. And we have a consistent performance. Like every time when the batch is released, it will have a consistent performance followed by a quality control. So how we will quality control the product? So there is so agrogen consists of a certain molecular weight of galactose-sulfur oligosaccharides that induces the maximum plant growth in the plant. So we have a chemical signature. After the extraction of those biostimulant, the batches are passed. Those batches will have a distinct chemical profile. We'll go for the bioassays. So we have a consistent product every single time. And then we have tried this. This product is sold more than 25 countries around the world, supported by around 40 plus institutional trials and 100 plus R&D trials. So minimum is 10% of increase reported in different crops, while the maximum in certain crop it goes to 20%. We see there is an increase of the yield. So now coming to the science of this product. So most of the product says when they are applied foliarly, it improves the foliar growth. But there is no scientific data for that one. So we have done an experiment. Actually, I had a video there. I don't know how to play it in the slide. Okay, leave it. We have performed an experiment where we use cotyledon, cucumber cotyledon as a system to show when we apply foliarly any product, it will improve the leaf area. So when we have observed that in the treatment. So in the lab, we use LB6 as a product code for the agrogen. So now often I call agrogen as an LB6 too. So you see there's an increase in the leaf area in the LB6 when it is treated in the leaf area of the cotyledon when it is treated with the LBS6, and that we can see by the leaf area too. So then next, what we have done to, because in the cotyledon, starch is the main source of the sugar that is available for the expansion. So we have shown when we had an cotyledon that is treated with an LBS6, it shows more amylase activity, means LBS6 treated cotyledons are better adapted to convert the starch to the sugar, sucrose, and that sucrose is used as a energy for the expansion of the cotyledon. And we have a higher amylase activity. We have quantified those things and to different sugars and shows that when we are treating it within LBS6, we have more amylase activity. To get it in the more of detailed molecular mechanism, how does it happening when we are applying a agrogane as a surface? How does it improves the foliar growth? So we we selected the certain genes that are involved in the cell division, proliferation, and the size. And we have found that when we have an agrogane, the starch, that is the sugar present in the cotyledon, is converted to the sugars by the activity of amylase, and that amylase is basically converted to that sugar is basically converted to the sucrose, and that sucrose is acting as a signaling molecules that regulate different gene expression. Basically, if I say this target of reformsin that is involved in the cell size is induced, and the genes that are involved in growth regulated factors are induced, and the genes that are involved in the cell expansion and cell proliferations are also induced by the LBS6 application. Additionally, most of the regulatory genes, we have found a gene called an expansion 1,5 that basically regulate the non-enzymatic expansion of the foliar part is also induced. Um, um, Along with the cell development genes, we have found this agrogane is differentially regulating the expression of genes involved in gibberellic acid biosynthesis by downregulating the genes called DELA proteins. So DELA is basically a downregulator of gibberellic acid, and its expression is less, and that is why we see more cell division and expansion rate. So overall, if I say agrogane has an so people will say, okay, you have shown on the cotyledon it is a very small bioassay, so that needs to be translated to the whole plant system. So we have done an hydroponic system where we sprayed the plants with an LBS6, and we see there is an increase in the foliage area, and that is also quantified, and we see a significant increase into the root biomass as well as the foliage area. And then we have done certain photosynthetic parameters, that is like linear electron transport and electrochemical shift and proton conductance as well as proton flux through. So with, if I want to brief it in a very simple way, so the plants which are treated with the agrogane, they are very efficiently converting the chlorophyll, very efficiently converting the photosynthetic energy to the ATP, in form of the ATP, which is used as the energy for the expansion of the cotyledon. Because so this is how we have a scientific backing when we are applying agrogane as a foliar spray. It improves the leaf area, and that because of the improve the leaf area, it can have more sunlight that helps in the more photosynthesis and more fixation of a carbon dioxide and more yield. So if I summarize this section, like it is a agrogane, we have shown that it improves the expansion of the leaves by regulating the cell division and proliferation, as well as it is very efficiently converting the storage sugar into the sugar that is used by the plant. And it is also involved in the regulation of the genes in phytohormone metabolism. Actually, I refrain because in the most of the literature, scientists have mentioned these biostimulant acts as a source of phytohormone, Rather than I say they don't act as a source of a phytohormone, but they induces plant natural process, plants natural metabolism, phytohormone metabolism that in turns helps the plants to grow. Again, so coming to the next stage, so not only this seaweed biostimulants has an impact on the plant growth. This seaweed biostimulants also plays a very major role in improving the soil health. How does they do? So for that, we have done a metagenomics experiment to just see what is an impact of the foliar spray of these products on the soil microbial diversity. So we have done conducted this experiment in the pot assay, soil assess, where we have a basal soil, and then we have treated the maize plant for around 28 days, twice spray. And then after that, we have collected the soil from the rhizospheric region uh, at a 2 time points. So what we have found, so we have found that this the maize plant by the foliar spray, we see there is a considerable increase into the morphological growth that we have shown before, and it improves significantly the number of the leaves, the greenness of the leaves, and total soluble sugar and secondary metabolites in the plant. But this is not my objective of this experiment. My objective is to see how the soil microbial diversity is changed. So here, if you see B, C, and D, so there is B is the microbial diversity of basically the bulk soil where we don't have any plant. So we can see that there is no purple area in that chronograph. But when we are adding a plant, we see there is a purple. So that purple area represents proteobacteria that are mainly influenced by the plant growth in the soil. Uh, Purple and this orange area both. And then when we are putting in LBS6, we are seeing that there is a reduction of the purple bacteria, but there is purple region, but there is an increase of the orange region. So orange region basically determines the gamma proteobacteria, while the purple region determines the alpha proteobacteria. So this graph shows that the application of LBS6 is helping the plant to attract the microbes that is beneficial for the plant growth rather than having all the microbial diversity. So the microbial diversity is kind of altered in a way so that plants have a more microbes that improves the plant growth. So in the soil treated with an LBS6, we find very high abundance of cyanobacteria, actinobacteria, firmicutes, microbiotes, acidobacteria, and mycocota. While so, and then what we try to do, we try to assess the function of those microbes in the soil based on KAG metabolism, and we find those the microbes that are enriched with certain metabolism are enriched by in the presence of an LBS6. So if I say an example of nitrate and sulfur metabolism, you see there is a certain red areas which shows that the bacteria that is involved for the nitrogen assimilation, they are more in an LBS6 soil as compared to the bulks in the control. While for the bacterial, if I say about bacterial chemotaxis, so the microbes has more chemotaxis is induced near the root zone, and so if I want to summarize everything in one picture, how is working? Basically, when we are applying an LBS6, it is altering the root exudates, and root exudates offers a carbohydrate that specifically that is helping the bacterial community to come and colonize the roots and help the plant growth. In that process, basically, in that alteration of the soil health and fertility, what LBS6 is doing sustainably, basically, it is improving the microbes that is having a high acid phosphatase, phosphorus metabolism. It is having the helping the plants to better plant defense, as well as the microbes that helps in nitrogen assimilation too. So, this is and the farm. In the last, that I want to acknowledge my mentors, basically, specifically Dr. Balakrishnan Prithviraj, who gives me an opportunity to understand the seaweed biostimulant, and then Alan. And Alan is like the guy who introduced me to the seaweed biology. 90% of people think that I still work for the seaweed cultivation. No, I just work on the seaweed products that is helping the plants to grow. My major area of work is on seaweed-based biostimulant for sustainable agriculture. But the Alan was the person who introduced me to the seaweed cultivation and seaweed biology people, and now I'm trying to learn that. Sri Shurnaran, he is the MD of C6, who always supports my ideas, and my PhD supervisor, Pradeep Kumar Agrawal. So here is my research team that comprises of Dr. Niveta and Dr. Deban. Niveta has a PhD from IRI, and she had done most of the work that is I have shown. And Deban is a part of the quality control team. And least not least, there's other colleagues, Shalja, Sawan, and Girish, who helped me to accomplish these experiments. Thank you, and I'm ready for any questions if you guys have.
Thank you, Dr. Shukla, for elaborating on C6's product Agrogain as a biostimulant.